Hereby I open this academic ceremony at Maastricht University. My name is Ton de Goeij. I am the pro-rector of the ceremony and chair of the committee. Welcome all members of the degree committee, in particular the 4 supervisors sitting in front of me. The first supervisor is Professor Jan Glats. He is professor of metabolic aspects of cardiovascular disease at Maastricht University. Dr. Miranda Nabben. She is assistant professor of heart metabolism at Maastricht University. Dr. Joost Luiken. He is associate professor of heart metabolism, also at Maastricht University. And Dr. Dietbert Neumann. He is associate professor of biochemistry of the heart muscle. I will introduce the 5 opponents later during the ceremony, because when I mention those names you will certainly forget. So I do that later during the opposition. Welcome the audience here in the aula, and I welcome also all the followers of the live stream. And I forgot to introduce the candidate. And our candidate today, how can I forget this? Our candidate today is Mrs. Myrthe Willemars, and she will defend her thesis in public, and her thesis is entitled Untangling the Threads of the Heart, Sex Differences in Cardiac Metabolism and the Role of Cardiac Hypertrophy. I wish you success in the coming hour, and I would like to invite you to give a summary of your thesis. The floor is yours.
Thank you, Pro-Rector. And good afternoon, everyone. Dear esteemed members of the Corona, dear colleagues, friends, and family, thank you all for taking the time to be here today, either on-site or online. I will gladly begin with a short presentation about my PhD research. And I would like to start that by drawing your attention to cardiovascular disease. This is a group of disorders that affect the heart and blood vessels. And you have probably heard about these diseases before, as they are the leading cause of death globally and pose a large economic burden on society. However, were you also aware that there are large sex and gender differences in cardiovascular disease? Historically, cardiovascular disease has been considered to be a male disease, as men used to develop cardiovascular disease at a younger age, in higher numbers, but also had worse clinical outcomes. As a result, research and therapy for cardiovascular disease has been completely tailored to men, which has significantly improved their outcomes, but has left women overlooked. Luckily, over the past decades, more attention has been given to cardiovascular disease in women. Since then, we have learned that women also develop cardiovascular disease in large numbers, but usually at an older age. Also, that currently more women die from cardiovascular disease than men, and that women are influenced by female-specific risk factors, such as pregnancy and menopause, but are also more susceptible to certain traditional risk factors that are shared by men and women, such as diabetes. We now also know that when women develop cardiovascular disease, their symptoms are usually different from those that are seen in men, which is why these have been labeled as atypical in the past. And finally, because cardiovascular disease medication and therapy have been optimized for men, we are now finding out that these are not always optimal for the female body. One form of cardiovascular disease where a clear sex difference can be seen is cardiac hypertrophy. When a healthy heart is exposed to triggers, such as stiffening of the heart valves or high blood pressure, the heart will have to pump harder in order to counteract this increased workload, which can result in a hypertrophic response in the heart. This hypertrophic heart is primarily characterized by a thickening of the heart wall, as visualized here. When taking a closer look, we see that this thickening of the heart wall is the result of cell growth, where the muscle cells of the heart become bigger in size. And while this has been considered to be an initially beneficial response, over time also negative effects can start to occur. For example, fibrosis, where the muscle cells of the heart are replaced by scar tissue. This will cause the pumping function of the heart to decline over time, until the heart's function is so impaired that it can even result in death. Then I would like to draw your attention to cardiac metabolism. As you know, your own heart is constantly beating, but in order to do this, it requires a lot of energy. Luckily, your heart is a metabolic omnivore, which means that it can use various sources to obtain this energy. The majority of this energy can be obtained from fatty acids and glucose, but also a small contribution can be made by ketone bodies, amino acids, and lactate. We know that in a healthy heart, there's a certain balance between this use of glucose and fatty acids for energy production. However, research has also shown that in a hypertrophied heart, this balance has been disturbed. With this in mind, a newer approach for therapies, known as metabolic therapies, are currently emerging. This newer form of therapy aims to restore the balance between glucose and fatty acid use in order to make the heart healthier again. At this point, I've introduced three main topics to you: cardiac hypertrophy, sex, and cardiac metabolism. It has been shown that there is a link between cardiac hypertrophy and sex, but also between cardiac hypertrophy and cardiac metabolism. In addition, there are indications for a possible connection between sex and cardiac metabolism. However, it remains unclear how these three parameters interact exactly. Therefore, the aim of my thesis was to investigate the role of sex in cardiac metabolism and hypertrophy, and to apply this newly acquired knowledge to the development of novel metabolic therapies. In order to get a better overview of the already available knowledge, I started with a literature review. For this review, I looked for the information for each combination of each of these three elements, and finally all three elements combined. In short, this review showed that there are sex differences in the structure and functioning of both the healthy and the hypertrophic heart, that there is a metabolic imbalance in cardiac hypertrophy with a disturbance towards relatively more glucose and less fatty acid use, that there are indications for sex-specific metabolism, where the female heart appears to use fatty acids more efficiently, and finally, for all three elements combined, that there are indeed almost no studies that have looked at the interaction of these three parameters altogether. The findings of this review have led us to a new hypothesis for sex-specific metabolic therapies. For this hypothesis, we propose that both the male and female heart have a metabolic disturbance in cardiac hypertrophy, but that because the female body appears to be more dependent on fatty acid use, that this disturbance is smaller in the female heart compared to the male heart. This would consequently mean that the female heart would only require a smaller correction by metabolic therapies compared to the male heart. While working on this review, there was something that I noticed. It appeared that all studies that investigated sex differences in the heart did so using whole animals, rather than just cardiomyocytes by themselves. The findings of these studies have led to the conclusion that there are sex differences in the heart, and that these are the result of sex differences between the behavior of male and female cardiomyocytes. However, upon closer inspection, it appears that this has never actually been examined in cardiomyocytes in isolation specifically. In addition, because in a whole animal the heart is still intact, it is still possible for the different cell types in the heart to interact with each other, or for the heart to even interact with other organs. This leaves the possibility that sex differences in the heart might still originate from a source other than the cardiomyocytes. So we decided to investigate these potential sex differences in cardiomyocyte metabolism and functioning directly into the isolated cardiomyocytes. To do this, we had male and female rats, from which we excised the hearts, and from these hearts we isolated the cardiomyocytes in order to obtain male and female cardiomyocyte cultures. These cultures were then compared for several parameters of interest, including their metabolism, their cell growth, their gene expression, so their genetic programming, and their contractile function, so how well these cells were able to beat. What we then found was, surprisingly, and in contrast to what literature is currently suggesting, that there were actually no distinct differences between the male and female cardiomyocytes. Now, sex differences might not only arise due to sex chromosomes or genetics, but also due to sex hormones. To evaluate the role of sex hormones, we exposed male and female cardiomyocytes to either estrogen, testosterone, or DHT, which is a stronger form of testosterone. However, also here, no profound reaction to any of these hormones in either the male or female cardiomyocytes was found. At this point, we did not find any sex differences in healthy cardiomyocytes and decided to continue with hypertrophic cardiomyocytes, where sex differences might still be present. To do this, we again had male and female cardiomyocyte cultures, to which we applied a hypertrophic stimulus to make them sick and hypertrophic, and then compared them for several of the previously mentioned parameters. Nevertheless, also in hypertrophic cardiomyocytes, no distinct sex differences were found. For the next part of this presentation, I would like to address metabolic rebalancing. In my thesis, we evaluated two different approaches to achieve this rebalancing. The first approach was by manipulation of endosomal pH. Endosomes are compartments within cells with a certain acidity, and by altering this acidity, we should be able to affect biology in a way that we can restore the balance between glucose and fatty acid use. To examine this approach, we had cardiomyocytes with a hypertrophic stimulus and exposed these to 4 different drugs that in various ways might affect endosomal pH. What we found was that 2 out of these 4 compounds were able to restore the metabolic balance, but that none of them were able to prevent the increase in cell growth. As this project is still ongoing, we've not yet been able to evaluate the effects of these compounds on other parameters, such as contractile function, but we still plan to do so in the future. Nevertheless, at this point, 2 out of these 4 compounds seem promising for further investigation. Then, for our second approach, we targeted the PI4K3 beta enzyme. This is an enzyme that is indirectly involved in the uptake of glucose. And by inhibiting this enzyme, we should be able to lower the amount of glucose that is taken up into these cells and thereby restore the metabolic balance. To investigate this approach, we used a similar setup where we had cardiomyocytes with a hypertrophic stimulus, and we exposed them to a drug that affects our PI4K3 beta enzyme. What we found was that this compound and approach were able to restore the metabolic balance, and while there was no positive effect on cell growth, we were able to restore the contractile function in these cells. This is a very important finding, as many of the currently used therapies for cardiac hypertrophy are aimed at reducing cell growth, while our findings are now indicating that this might not be an absolute requirement. Just by targeting and restoring metabolism alone, it would be possible to restore the contractile function in these cells. In addition, I would like to point out that this approach was successful in both a preventative setting, where we prevented the cells from malfunctioning, but also in a treatment setting, where cells that were already sick could be made better again. To conclude, there are 3 main findings that I would like to summarize. First, that the research in my thesis did not find any distinct sex differences in either healthy or hypertrophied isolated cardiomyocytes. Second, that my thesis research found no effects of sex hormones in isolated cardiomyocytes. And taken together, these 2 findings suggest that sex differences in the heart might originate from a source other than the cardiomyocytes, but also that when studying sex differences, one should do so at a whole heart or a whole organism level. Then third, that metabolic rebalancing through endosomal pH and PI4K3 beta is a promising therapeutic approach, which is therefore suitable for further investigation and development into actual therapies. These findings help improve our understanding of how sex influences cardiac metabolism and hypertrophy, and unraveling of these mechanisms will help support the development of sex-specific metabolic therapies, which will ultimately improve clinical outcomes for both men and women. With that, I thank you for your attention, and I give the word back to the prorector.
Thank you very much, Mrs. Willemars, for a clear presentation. The opposition will be opened by the chair of the Thesis Assessment Committee, Professor Janine Prompers. She is Professor of Organ-Specific Metabolic Imaging at Maastricht University. Professor Prompers.
Dear candidate, I've read your thesis with great interest, and I would like to congratulate you and also your promotion team with this extensive amount of work. Your thesis addresses an essential gap in cardiovascular research, namely that actually at the moment we don't know enough about sex-specific differences in cardiac disease mechanisms and how to properly treat female patients with cardiac disease. You've shown to be also a very creative person. I was really impressed by the crochet that you have demonstrated in your thesis and actually there in front of you at the desk. I've never gotten any further than a simple pillowcase, but yeah, I'm really impressed by this. But of course, I'm also here to ask some questions, and I would like to ask you about chapter 4, where you explore the role of sex in cardiomyocyte physiology. And you show that at the whole heart level, you do see some differences in, for example, heart size between male and female rats, and also a small difference in the expression of the fatty acid transporter CD36. But where it is actually slightly increased in the female heart. But when you then look at isolated cardiomyocytes, you do not observe a difference in CD36 expression. And also, when you look at fatty acid uptake, this is equal among male and female cardiomyocytes. And for me, as a MRI person, this is really confusing. So could you explain a bit further how I should interpret these different results at the whole heart level versus the cardiomyocytes? And I was wondering, yeah, do other cells in the heart then also express CD36, or how should I look at these differences?
Mm-hmm. Highly esteemed opponent, thank you for your question and your compliments regarding my thesis, and also for acknowledging my crochet pieces. It is indeed true that in my thesis we show that at the whole heart level we see a higher expression of CD36 in females rather not in male, while at the isolated cardiomyocyte level this is equal. Of course, the difference between those two samples is that in a whole heart, we don't only have the cardiomyocytes, but also other cell types, like fibroblasts, endothelial cells, maybe also some inflammatory cells. And I do indeed think that there are no differences at the cardiomyocyte level. So this higher expression that we see in a whole heart has to come from these other cell types. And it is indeed known that endothelial cells also express CD36 because nutrients, fatty acids, are coming from the bloodstream through endothelial cells and thereby go into the cardiomyocytes. So what I suspect is that females have a higher CD36 expression in their endothelial cells, and that might also give rise to potential sex differences and like fatty acid metabolism eventually. So that links back to the hypothesis in chapter 8.
Okay, yeah. But then in your discussion, you also mentioned that the fact that you do see a difference at the whole heart level and not at the cardiomyocyte level is that would be an interaction with other cardiac cell types. Can you explain a bit further what you mean with that?
So the idea with that is that intrinsically, if you take cardiomyocytes like in isolation, in carotene, like just by themselves, the way that they naturally behave, what we have observed that if you place them in this like environment, this situation, the male and female cardiomyocyte act exactly the same. So what I suspect, as a way to still confirm the findings that I found, but also make them like in compliance with literature, what are suggested, is that other cell types in the heart, so the fibroblasts or the endothelial cells, that they might still be intrinsically different, that they by themselves still express sex differences or are influenced by sex hormones, which makes those cell types act differently between the male and the female cells, but that the effect that they, like the interactions that they then have with the cardiomyocytes, conveys a way of how they like direct the cardiomyocytes to behave in a sense. So you could compare that like to saying you have a group of people, if you put someone alone in a room, they will act differently than when you put them in a party or when you put them like in this academic setting. People will behave differently based on the influences that they have around them.
And how fast would you lose that effect? Because I think you cultured your cardiomyocytes for 24 hours or something. Is that then sufficiently long to lose all those effects, or?
Apparently it might be, but the functional analysis that we have done in chapter 4 have been done after 24 hours of culturing, while more of the Western blotting, like the protein assessments, those samples have been come directly from the isolation, so only a few hours.
Okay.
And apparently either differences are lost or, we need to do further investigation to really say what is what.
Okay, clear. And then, of course, one of the things to investigate was then the effects of sex hormones, which you also did a lot of experiments, and surprisingly no effect at all. So I was wondering, can it, because you mentioned other cells in the heart, but could it also be other systemic effects? So in chapter 2, for example, you show that there are sex differences in energy metabolism also in other metabolic organs like the fat tissue and the muscle tissue. So that could perhaps lead to other systemic effects like differences in nutrient supply. Can you elaborate a bit on that, how that would affect?
Yeah, no, it is indeed very possible because the model that we've now used, you have just plain cardiomyocytes, you don't have any hemodynamics or like nutrient supply that might be different. So that is like something this model does not take into account. If you look at the two like hypothesis directions in chapter 8, it's either that through interactions with other cell types, the cardiomyocytes are made to behave differently. That is one option. Another option might be that the cells, like say the engine is the same, but the energy, the fuel that they're getting might be different. And we see indeed that other organs in the female body, like the skeletal muscle, adipose tissue, is all fine-tuned to fatty acid metabolism, which will also mean that the supply that the heart gets will also be different in the female than in the male. And because that supply is different, that might make the cardiomyocytes adapt to that supply.
Yeah. But then if we know, so how much do we know about the differences in supply between males and females? And should you then design your experiments in a different way? Because now the cardiomyocytes are all cultured in the same conditions, right? But would you then maybe have to change that? Or
we already know from people that have looked more into like diabetic cardiomyopathy that if you apply a different nutrient supply, so for instance a high-fat diet, the cardiomyocytes do react to that. They do change their metabolism, so that concept in itself has already been proven. To really investigate like how different nutrient supply between male and female would affect the cardiomyocytes, you would first need to know how that differs. At this point, that is quite understudied, I would say. I've tried to look for it. I found a couple studies that indicated, okay, there might be some differences in these proteins, lipids. But the tricky thing with that is that you need very standardized conditions. Like, do you do it with fasting? Do you do it without fasting? And from what I've read, that makes it quite complicated.
Yeah.
And also, a lot of studies focus, of course, on diseased states and not on healthy individuals, which is what you would want to know first.
Yeah. Yeah, I guess this is challenging with models in a general sense, right? Because so then you could go to so cardiomyocytes is a certain model, whole heart is a model, but then you could also go to a whole animal. But of course, the best model of a human being is a human being. So if you would have the chance to study this in humans, what would you do? So at the whole human being level.
Well, ideally, we would like to characterize cardiac metabolism indeed in humans. You could indeed use non-invasive imaging techniques with radioactive tracers, really characterize how the female and male heart differ in glucose and fatty acid use. Let me start over. No, we can indeed use metabolic imaging in vivo, because in the heart of a human, we of course can only do non-invasive analysis. So if we can use non-invasive metabolic imaging to characterize how the male and the female heart would use fatty acids and glucose in both the healthy state and in the diseased state, you can monitor how that might change over time. But it might also tell you more of how it functions.
Yeah. Well, thank you very much for your answers, and I give the word back to the pro-rector.
Thank you, Professor Pompers. The next opponent is Professor René Ventura-Clapier. She is Professor of Cardiovascular Pathophysiology, and she is affiliated with the Université Paris-Saclay à Paris, France.
I would like to thank you to invite me for your defense, because this is a really subject that is very important for me. And I really appreciate your results, because nobody was able to differentiate what is from the organ on the whole body and what is coming from the isolated cardiomyocytes. And I think that your results are quite challenging. Even if I consider that you, of course, didn't investigate all the different functions and different energy involved in energy metabolism, than, for example, mitochondria, which are known to be quite different between male and females, and I would be very interested to see in your model whether you can evidence this or not. But voila. So I think that your work is also an important guide for future researchers. And the discussion is very, in fact, informative and of interesting and big value. And especially the discussion we had for the possible interaction between different cell types and cardiomyocytes. So I think this opens large avenues for next experiments and next young scientists. What I wanted to ask you, so now my main concern, or not concern, but I would like you to explain in quite details the basis of your reasoning on the role of, for example, glucose metabolism, glucose uptake in view of fatty acid uptake, and what link you are doing between glucose uptake and protein synthesis, hypertrophy, and how you think that you can counteract hypertrophy with glucose metabolism. I would like you to explain what are the different steps of reasoning on this basis.
Mm-hmm. Highly esteemed opponent, thank you for your compliments and appreciation for my research. Let me get a moment to gather my thoughts, because we know that there are indications of like pathway connections between glucose uptake and also hypertrophy. We have different ways that glucose uptake can be stimulated, like insulin-stimulated glucose uptake. We have certain pathways that induce GLUT4 translocation to the membrane, increase glucose uptake. We also have other pathways that are more contraction-stimulated, where you have proteins that are at a crossroads between stimulating glucose uptake, but also hypertrophic signaling. And we suspect that in case of these diseases, there is an activation of that pathway that would therefore activate both directions. Does that answer your question, or did I misunderstand?
You really think that you can, by changing glucose metabolism, you can change hypertrophic signaling?
Well, in the findings that we now have, we only looked at 24 hours. There we did not see any significant changes, but we do see in chapter 7 some indications that there might be some positive feedback loops, that if you would culture these cells longer and evaluate them over longer time, that also over time hypertrophy might be restored. And that is then also via the pathways that I tried to address. That there appears some kind of feedback mechanism that if we restore the glucose uptake, eventually that pathway gets downregulated, but therefore also the hypertrophy eventually goes down. And that would be very beneficial, because then you still have both aspects that you would want to restore achieved.
I have also a comment, because you are always speaking about hypertrophy. You have your model with phenylephrine stimulation, but in fact you don't see any hypertrophy, and it is quite normal, because 24 hours may be not sufficient to have a real increase in protein content and really increase in the size of the cardiomyocytes. So why didn't you use hypertrophied hearts taken from whole animals with hypertrophy and then compare this isolated cardiomyocyte from hypertrophied animals?
Mm-hmm. It is indeed possible to also use cardiomyocytes from hypertrophy in vivo models. The reason why we have used this phenylephrine model is because a previous colleague of mine has dedicated a lot of our work to that, to characterize that. We still see that it ticks off all the check marks of like what a hypertrophy model should have, so the change in metabolic balance. We do still see some increases in protein synthesis. So we do have some indications for hypertrophy, that also hypertrophic signaling is increased, the hypertrophic gene expression is up. We see contractile dysfunction. But also from a practical point of view, this would be easier than using in vivo models, and also from ethical considerations. If you use living animals, you have to take their well-being into account. While if you would use phenylephrine model, we can have healthy animals that are only euthanized and have less discomfort. So there's also considerations that you have to take along.
But using animals, you can also dissociate between adaptive hypertrophy and maladaptive hypertrophy. And this would be interesting to study in terms of sex differences also. Mm-hmm.
Yeah, for this phenylephrine model, the way that I understood is that we're looking more at a compensated phase of hypertrophy still, rather than the more advanced direction into heart failure. But it is indeed the case that if you would look in vivo models, you can look at more specific stages. That is true.
And you couldn't keep your isolated cardiomyocytes for more than 24 hours?
Yes, we can keep them happy and healthy for 48 hours, but we could keep them alive longer, but then we had to add additional metabolic compounds that are also going to influence metabolism, which is what we want to study. So that is something that for our specific research is not feasible.
Because when you look at, at your results, you see change. You can influence, for example, with the inhibitor, you can influence glucose transport, but you don't influence signaling of hypertrophy, for example. So it means that maybe it's too short exposure, or why would you explain this?
I would think that indeed if we could keep these cells alive for more than 2-3 days, then over time we might also see, after correcting cardiac metabolism, also a reduction in the hypertrophic growth. At this point, it's not completely clear yet from, I think, what exactly the mechanism, the detailed correlation of connection between glucose metabolism and hypertrophy is, but there are very strong indications for it.
If I have another question from experimental point of view, you seem to have some cautious with statistics. From your propositions, you seem to have some ambiguity with the statistics. So I would like to know exactly which statistic you use and why did you use them, and in which circumstances. And I think I have maybe one problem is that very often you have very small number of samples. And I think this is a pity because sometimes, for example, for some of the experiments, it seems that if you have more samples, you would have really statistical differences between male and females, for example. When you do like complex 1 experiments, or also other phosphorylated CAM kinase 2, for example, it seems that really you are not far from significance. Mm-hmm. So I would like to have your comments on this and on statistical experiments.
Yeah, I agree.
Analysis.
It would indeed have been nicer if we could have increased our sample sizes higher than what they are now. Fortunately, the time did not allow for me to do that, but for publication, we are still planning to increase our sample sizes. Indeed, we lose a bit of power from that, and therefore some things that might be significant are now not yet. For the statistics that have been applied, the reason why I included this proposition is because over time I've talked to a lot of different people about what would you advise me to use for these statistics, and it's striking that everyone gives a different answer and seems also quite opinionated about it. And at some point I thought, okay, I will just talk to the statisticians at the university and get information and advice directly from the source people that studied for this. And the proposition that is in my thesis is actually something that one of those statisticians told me, that if you asked 2 of the statisticians for advice, you will get 3 different answers. And I think that nicely highlights that, yes, there is a very clear white area of what is good in statistics, some black area of what is absolutely false, but there's also still a gray area that is potentially up for discussion. But as long as something like your statistical approach makes sense, you can you can reason why this has been applied, then
Okay, thank you very much.
Thank you, Professor Ventura. The next opponent is Dr. Vanessa van Empel. She is Associate Professor in Cardiology at Maastricht University. Dr. van Empel.
So, dear candidate, first I would also like to congratulate you on the completion of this very extensive thesis. Very impressive. I would also like to include your promoters and co-promoters in these congratulations. I think sex difference is something that's very hot topic at the moment, and I think it's overdue for a long, long time. So I'm very interested that you studied cardiac metabolism and sex differences in that. I would also like to ask your opinion a little bit more about Chapter 4. You just discussed that a bit with Professor Prombers, but I would like to still look more into the details, because I'm also surprised at why you see differences between sexes in whole hearts and not in isolated cardiomyocytes. And then if I look at figure 4.3, I wonder if it's because the males just have more cardiomyocytes compared to the females. You look at the yield that you have from the cardiomyocytes and you see, oh, that's just much more cardiomyocytes in male hearts compared to female hearts. Do you think that's something that would explain differences between men and women?
Esteemed opponent, thank you for your question and your compliments. Yes, from the cardiomyocytes that we isolated from these rat hearts, we indeed see that the male heart is bigger and that also the yield that we get from these isolations is higher. Of course, there's something that we have to take into account, that this is still a technical procedure, and we had a lot of experience with this technique in male hearts, but not in female hearts. I was the first one to start with this. What we've also seen is that over time, the yield that we got for the female heart improved as we optimized the method for this. So that is something that in the back of our minds we have to take along. But if I address your question...
Why would you expect a difference between male and female hearts with isolation cells?
Sorry?
Why would you expect a difference between male and female hearts with isolating cells?
Because the male heart is bigger. And we would indeed, the male, it has been shown in studies that the male heart indeed has more cardiomyocytes, but if you look as a percentage of the contribution of different cell types in the heart, we see that actually the female heart has a higher percentage of cardiomyocytes than the male heart. Mm-hmm. So that would not necessarily be...
so more cells per amount of gram of heart tissue. Do I understand that correctly?
No, because the total size of the male heart is bigger, then in total you would also have more cardiomyocytes. But if you look at all the cells in the heart, normalize it to the total amount, then we see that percentually, female heart actually has more cardiomyocytes than the male heart. So the male heart contains more additional other cell types, like for instance fibroblasts, endothelial cells, etc.
I understand. Do you know anything about the effect of aging on this amount of cells?
Amount of aging. Ooh. In a healthy situation or a diseased situation?
Let's talk about healthy aging.
I do know that there are a lot of changes during the heart's lifetime, and that it also differs between males and females, expression and fibrotic, collagen, etc. It changes a lot over lifetime, but specifically for the cardiomyocytes, I do not recall at this moment.
No. And I was also wondering, because I wasn't sure, this is an adult cardiomyocyte, but for female adults, it's also very important if you look at the heart, if it's premenopausal or postmenopausal. Mm-hmm. I work with humans, so I don't know anything about all these animal models. Is this premenopausal hearts or postmenopausal?
I would consider this a premenopausal. These animals were at the age of 3 to 5 months, and rats only go into equivalent of menopause at around 9 months. So I would consider this premenopausal. Yeah.
So then it's probably not that surprising that we see just little differences and even less differences in isolated cardiomyocyte, because in young adults, male and females are not that different. It's mostly as they age that we see, and especially postmenopausal, that we see so many differences, and especially in response to certain stresses. Mm-hmm. Especially if males are protected for a lot of cardiovascular disease until they're postmenopausal. Do you think that factor that the hormones protect your heart, protect maybe the cardiomyocytes, is something that you can incorporate in these experiments? Or just thinking, if we would study menopause, what could we do for additional experiments? Now that we have your data, what could we do going forward?
You could try and mimic menopause maybe in the rats, but then still, if you take the cardiomyocytes out of isolation, in isolation, it might again be that there is no difference. If it is indeed the case that cardiomyocytes by themselves are not different, then I think also if you would use a postmenopausal model, you might not find anything. The influence of sex hormones might still be tricky, because also here in the healthy cells, we're not finding any response. And I would expect that to be the same in both the pre- and postmenopausal cells, so to say.
Yeah, I wonder about that. I'm not that pessimistic that you don't see difference between pre- and postmenopausal cardiomyocytes. And I think that's why it's also interesting that you look in chapter 5, where you also stress the cardiomyocytes in hypertrophy, because that's something that we see in everyday clinic, that healthy people are not that different, but in response to stress, in response to smoking, in response to other things, then is where the differences are more pronounced. So even if you have postmenopausal women who are not protected by the hormones anymore, maybe an extra stressor will also see differences in cardiomyocytes. But something else, like I said, I'm a clinical cardiologist, so I was quite interested that you have some human data as well in figure 4.8. But first I thought, oh, these are human biopsies, but then I saw, oh, they're human cardiomyocytes. How do you get data from human cardiomyocytes?
Yes. So this data from figure 4.8, this is a data set that we combined from previously pre-existing data sets. So other researchers have already collected biopsies from healthy hearts, maybe post-mortem, like from people that have died from motorcycle accidents. And they have used these samples and done single nuclear RNA sequencing on them, on both these healthy samples and a diseased setting, and also had that as a primary research question. But these data sets are freely available online. And of course, those previous studies had a completely different research aim. They really want to see what is the difference in this disease, while we wanted to see, okay, can we use this data set, use it to compare healthy male and females, because indeed, healthy material is very scarce, let alone get it for male and female in high numbers.
And then you say that there's already diseased patients that have post-mortem samples, are these?
Yeah, healthy individuals, but that have died from unfortunate circumstances.
Yeah. And do you know the age of the females?
From the females, we have looked at the age. It was a very broad range, so we were unfortunately not able to distinguish pre- and postmenopausal. We really wanted to do that, but unfortunately then the sample sizes would still have been too low. So we had to keep it like a whole range of 18 to, I think 80 off the top of my head, like a broad range.
But all probably still premenopausal women though?
Most of them.
Yeah.
I would have to revise to be absolutely sure, but yeah.
You're right, it's very difficult to get healthy human samples, but maybe diseased human samples. Do we know anything about sex differences there?
You may give a short answer, please.
To give a concise answer to that, you would really have to address a specific pathology, but we do really see sex differences also in different pathologies, like HFrEF, HFpEF, etc. And I'm happy to confer, to discuss this further in the reception.
That's good. All right.
Thank you, Dr. Van Empel. The next opponent is Dr. Martijn Hoes. He is Assistant Professor of Cardiology at Maastricht University. Dr. Hoes.
Thank you very much. Before we go to my questions, I would like to congratulate you on this achievement and this very nice thesis. Not only have you made a substantial contribution to the science behind the work, but you've also demonstrated that dedication and hard work actually leads to something very successful. Let me see. So, actually, I would also like to extend my congratulations to your promotion team, your friends and family that have made this all possible by supporting you in the meantime. And before I would like to go to chapter 5, I would be mostly interested in how you look at hypertrophy in general and how that will actually be a disease of the heart or the cardiovascular system that we should worry about. Can you speculate some about that?
Esteemed opponents, thank you for your compliments from me and the promotion team, and also your question. Are you then asking me, just to clarify, are you more asking is it a disease of the muscle cells or is it a disease of the vasculature?
Actually, everything combined, but what I'm mostly looking for is an answer to the question, should we actually be worried about hypertrophy in the cardiovascular system? Because you've studied this in quite some detail, and it sounds very scary if you talk about all these numbers of mortality in both sexes, But should we be worried in general?
Okay. To answer that question, I would first like to bring up the fact that cardiac hypertrophy doesn't necessarily have to be a bad thing, because we can distinguish between both physiological, which is the good form of cardiac hypertrophy, and pathological hypertrophy. The good one we see in postnatal growth, compensations during pregnancy. The pathological one is indeed the one where we see negative effects such as the fibrosis and the cell death, and that is the form that we want to avoid. And if we then look specifically at the role of the vasculature, we see that in if you have proper angiogenesis, so construction of your blood vessels while your heart muscle grows, then this supports physiological hypertrophy, so the good kind. While if your angiogenesis is insufficient, you get problems and you develop more the pathological hypertrophy.
So it's not as black and white as...
Not necessarily. Well, normally when we talk about cardiac hypertrophy, we automatically go for the bad type, but we shouldn't forget that there's also a good kind.
Yeah, right. It's very beneficial to have the good kind, actually.
Yeah, for exercise, definitely.
I would like to invite you to brainstorm a bit about chapter 5 in this case, because you rightly conclude that there is no sex differences found in the isolated cardiomyocytes from rats. But you do mention that they were found in vivo models. So there is something going on. There's some difference in transitioning from an in vivo model to something that is in a dish, correct? Could you speculate why the effects are so small in your model compared to what you clearly show in an in vivo model in your reviews?
Then I would again have to refer you to the hypothesis of chapter 8, because if we take these cardiomyocytes in isolation, we are missing crucial interactions with other cell types, but also the function that they display within the heart. We know that there are indeed interactions between different cell types and the non-cardiomyocytes and the cardiomyocytes, and that they are close interaction. So if something goes wrong in a whole heart, we will have like a kind of snowball effect where things go wrong in one cell type, that conveys it onto the next cell type, it goes wrong in the other cell type. While with the cardiomyocytes in isolation, it stops with them. And
so regardless of what is actually the hypothesis of why you don't see any differences, I was wondering how important it is to actually see these differences in your models. If you look at the study at large, how big should you preferably like to see those differences in hypertrophy between the two conditions?
Wait, can you re-specify? I'm not completely sure I understand the question. Could you please rephrase?
Sure, of course. So you're trying to find a method to induce, I hope, highly significant levels of hypertrophy, right, as the basis of your model. So how large should those differences be to actually qualify as hypertrophy in isolated rat cardiomyocytes?
Okay, I see. We have used this phenylephrine model, which has been previously used also in other studies, also by my predecessors. We see that this indeed, as I addressed with Professor Ventura, that it checks a lot of the boxes that we want to see with cardiac hypertrophy. The thing, of course, that we have with modeling diseases is that we see something in the human. We might not completely understand how it works, but the things that we do understand, we try to model. So we're basically making a copy of a copy, and we hope that that is sufficient. Of course, it will not always be completely the same, exactly the same, because then it wouldn't be a model anymore. So there will always be some shortcomings, but we try to achieve as much like a representative setting as we can.
Yeah. I'm very happy that you mentioned the copy of a copy and you're trying to see something. And this was exactly what I would like to hear in this case. So I'm very happy for that. So by extension, if you use a rat model that is definitely not a human in any sense, how much would you expect from rats to actually develop hypertrophy?
We do know that the pathophysiological mechanisms in rodents do have some differences compared to humans, that it doesn't always translate as much as we would like to. But of course, we are limited by the options that we have. Of course, we can go to stem cells. But also because we're now looking specifically at metabolism, we would also want to have human cardiomyocytes, like iPSC-derived cardiomyocytes or other cell types that have a representative way of acting, phenotype. And sometimes science is still lacking a little bit in that. But also for sex differences, that might have been very tricky, because if you want to have a perfect isogenic control of male and female stem cells, at this point we're not able to achieve that yet. I do have some ideas to make them, but unfortunately science is not there to do that yet.
I see why you would go to taking stem cells into your answers. However, I'm not entirely convinced stem cells would be the right way to go. So for that, do you have any ideas of what the ideal model should be in this case? Because rats are not humans. They might respond differently to stimuli that would induce hypertrophy. Should you go to a different model if you were given the chance to do so?
We do know that the pig heart and the dog heart are more resemblant, but that of course comes with more ethical considerations, because by the 3R principles we always try to use species of the lowest phylogenetic, I think is the correct word, level in which we can still properly achieve our goals. So even though those models would be more representative, it also comes again with other challenges. Would
you like to introduce some genetic defects as well? Because you're now looking into just chemically induced hypertrophy. I'm not sure how relevant that is to the human situation to chemically induce it. We might actually be looking at hypertrophic cardiomyopathy that is almost always induced by a genetic defect, whether we know it or not. Might be polygenic, might be monogenic. Would you expand your model in that area as well?
Yes. So for this thesis, we have specifically looked into the phenylephrine model. If we would have had more time, we would have also liked to look at more naturally occurring, natural ways to induce cardiac hypertrophy, such as stretch models, where you constantly stretch your cells on a silicone membrane that more represents the pressures that the heart is exposed, like the cardiomyocytes in the heart are exposed to in an in vivo setting. That would be a more natural option. You could indeed also look more into the genetic underlying cardiac hypertrophies, hypertrophic cardiomyopathy, but then I think, and you should separate them until we know more about the non-genetic ones.
Excellent. Thank you very much. And with that, I would like to end my turn.
Thank you, Dr. Hoes. The fifth and last opponent is Dr. Jos Broers. He is Associate Professor of Genetics and Cell Biology at Maastricht University. Dr. Broers.
Okay, thank you very much. First of all, the candidate, congratulations with the book. It looks great. Also, congratulations to the promotion team. Looking at your wonderful cover, I noticed that you also have also not skills only in baking, but also in making these things. So she's let's say, organizing baking for the sake of the Dutch Heart Foundation. So, and then she bakes cookies with cell components on it, etc. Also, as a master's student, already have shown that in my group, you have shown that you are very capable of doing very well organized experiments. So you thought of all the things that were needed. So I can congratulate you with that. It was a shame that I could not offer you position then, because, it would have been very nice. But I was very happy that I gave you a good grade, because I saw you later kicking in the taekwondo studio. And her legs come up really very high. Okay, but coming to my question in chapter 4, I'm also, like other people, struggling with the fact that you do not see any effects on let's say, between male and female cardiomyocyte. And then, a kind of a technical question. How do you making sure that your sex hormones actually reach your cell interior? Did you check for that?
Esteemed opponent, thank you for your compliments and your question. The sex hormone part of the study has indeed been quite challenging. We have tried to look for various ways to confirm that the methods that we are applying are indeed correct. So to put it simply, is this a response that I'm supposed to find, or is it something that I am doing wrong technically? I have talked to many different people that use sex hormones in their studies, some of them in cardiomyocytes, some in then other cell types like hepatocytes. They have all confirmed to me that it should in theory be super simple. You don't have to use any specific requirements like protected from light or other things that you should keep in mind. And they were very surprised that I did not see any differences. So that is one part of my answer. The second part, we have tried to take along also positive control cells. So while isolating these cardiomyocytes, we also took along endometrial stromal cells from the uterus from the female animal and Sertoli cells from the testes of the male animal. Fortunately, we were not very skilled in those assays yet, so we could get some cells, but not enough that we could use them for experiments. So that was something that we wanted to try, but unfortunately we were not able to. So to come to a concrete answer, we've not been able to explicitly confirm that it reaches the cells, but we are quite sure that they should.
Okay, and did you consider things like perfusion of your cells with constantly flowing medium going into the system?
To the cardiomyocytes or in the whole heart?
In the isolated cardiomyocytes. So just pump in medium constantly rather than just dropping on top of the cell? No.
Okay, so the way that we have applied these hormones is that we just made a batch of medium to which the hormones have already been applied and then replaced the entire medium. So it's a bit different from continuously perfusing them, but it's not like we had medium, we drop it, and then it might just stick to the wall of the thing. So it should have been able to reach the cardiomyocytes.
Okay.
And also, if you look in literature, other ways that people have applied this, they also just say add it to the culture medium, it should be okay.
Okay, yeah. Okay, thank you very much. Then I have an additional question. So you mentioned that you did not find any estrogen or sex hormone receptors in the cardiomyocytes. Yet other people did show the presence of these receptors. Did you, for instance, also look at the GPER receptor? Mm-hmm. Did you investigate that?
Yes, indeed, if you look in literature, you find that a lot of people state that they have these receptors. I have not explicitly seen the articles where they have tried to assess, like, do they have these receptors, yes or no? But we have seen from the RNA sequencing that the expression of them was very, very low. Of course, you have to take into account that this method might have a low sensitivity, so I would like to back that up with qPCR, which is much more sensitive to assess like what is the gene expression of these receptors. But because we didn't see any differences, we also decided to look at the protein expression of these receptors using Western blotting. So we blotted the androgen receptor, estrogen receptor alpha, beta, and also the G protein coupled estrogen receptor. But the problem that we encountered there is that the high-quality antibodies from Abcan that have already been knockout validated gave us still so many bands that we started to refer to them as barcodes, which is not good. Also, not that there might be some pattern of post-translational modifications because the variation in height is just too big, also for like monomers, polymers to occur. It was very random. And that led us to like a technical limitation. Like if we don't have adequate antibodies to assess this, then yeah, we get stuck. In addition, what I found interesting to follow up with that is that for the antibodies that we use for estrogen receptor alpha and beta, we chose those because they have previously been used in tissue sections of female heart, but then using immunofluorescence. And of course, with immunofluorescence, you will, even if it's aspecific, you will find a signal, and people still think, if they don't properly compare it, I have a signal, I have the expression of this protein, while it might not actually be there. So I would have to actually concretely check up on that, but it might be possible that also these statements of is there expression of these receptors in cardiomyocytes might have been a misguided conclusion.
Okay, thank you very much. Right, then I have a final question. So if you look at the data, so suppose I assume that indeed, let's say the hormones did reach the cells. I did not see a difference in response or any response. Can you consider that there is something like biologically different male and female cardiomyocytes?
Yeah, that is something that has been brought up before. Yeah, I think it would depend on like how do you define that different. Like in They, of course, have differences in their genome because females have XX chromosomes, male have XY patterns.
Yeah, but would that have impact on the differentiation of cardiomyocytes?
On differentiation.
Is there any clue that, let's say, you will have different type of cells because of that?
I would have to look it up to be completely sure, but I do.
Okay.
Mrs. Willemars, the time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis and in particular the quality of your defense. And I request you to remain here for our return here later.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Mrs. Myrthe Willemars. The degree committee here present has assessed the quality of your thesis and in particular the quality of your defense. And in view of its positive verdict and of course taking into account the examinations that previously have been done by you, she has decided to grant you the degree of doctor. And Professor Glatz is authorized to confer upon you this academic distinction in accordance with Dutch university custom and law. I invite now your supervisor to take the floor. Professor Glatz.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Myrthe Maria Anna Willemars, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university. Dear Dr. Willemars, it's a great honor for me to congratulate you on this degree that you obtained today. You did a fantastic job, especially here in the defense of your thesis today. But the person from our team that was most closely with the supervision is Miranda Nabben, so I hand over to her to give the laudatio.
Hey, thank you. Well, I'm gonna read it from the paper because I don't dare to do it like by heart or like. But first of all, of course, my congratulations because I'm really proud at the way you did this defense and all the preparations beforehand. So you did an amazing job, I think. So I wrote, geachte Dr. Willemars, because we can say that now. Beste Myrthe, ook ik wil je van harte feliciteren met het behalen van je dokterstitel. Een geweldige prestatie waar je enorm trots op mag zijn. Vandaag is een heel bijzonder moment. En zoals gebruikelijk wil ik graag met jou even terugkijken en met al de anderen hier even terugkijken op de afgelopen jaren. Ja, het was ongeveer 4,5 jaar geleden dat je bij ons begon aan jouw promotietraject. En ik had net een behoorlijk uitdagend project van de Hartstichting binnengehaald, waar jij op mocht beginnen. We waren dus in eerste instantie op zoek naar een AIO. En ik hoorde vanuit de microscopiegroep en ook vanuit de zebravisgroep dat zij een hele goede stagiaire hadden gehad. Die zij dachten dat die zeer geschikt zou zijn voor dit project. En je was cum laude afgestudeerd met deze indrukwekkende onderzoeksstages en referenties voor die stages. Ook gerelateerd aan het LMNA-project wat je gedaan had. Dus wij dachten, dat gaat hem worden. En al snel bleek ook dat hun aanbeveling volledig terecht was. Ja, en er was ook, toen je startte, je maakte je de technieken eigenlijk razendsnel eigen. En je wist ook al heel vroeg in je promotietraject een eigen beurs binnen te halen. Een fellowship over gender en sex differences. En dat was een beurs van Zon en Wee, dus dat was al heel netjes gedaan. En die had ze vooral zelf geschreven, daar hoefden wij bijna niks aan te doen. Dus die haalden ze zo even meteen binnen. Dus dat was super natuurlijk. Maar dan ook naast je werk in het lab nog daarnaast en de manuscripten. En het viel ons ook al gauw op dat als er een SOP geschreven moest worden, want je hebt ook een aantal SOPs, nou dat was helemaal perfect in detail, echt helemaal goed uitgewerkt. Daar konden we Myrthe voor 100% op vertrouwen, met het RNL ook en zo. Je hebt het allemaal echt perfect uitgewerkt. Dus dat zagen we al heel snel dat je daar konden we volledig van jou op aan. En je liet ook keer op keer zien hoe zorgvuldig en gestructureerd je eigenlijk te werk kon gaan. Maar het was natuurlijk niet altijd even makkelijk, zoals we weten. Ik zei het altijd, dat hoort erbij een promotietraject. Maar als je erin zit, is dat niet altijd even makkelijk. En vooral met jouw project, wat nogal uitdagend was. Ook omdat we naar die geslachtsverschillen die we vooraf eigenlijk hadden verwacht, die vonden we niet terug. Dus dat was even nogal pittig. En uiteindelijk bleken die geslachtsverschillen helemaal niet te bestaan. En in het begin dachten we nog, we doen iets fout of wat dan ook. Maar het bleek gewoon echt niet zo te zijn. Of ze bleken er gewoon niet te zijn. En dat was echt eigenlijk een hele belangrijke bevinding. Des te belangrijker. Dus ja, onze hypothese die hield geen stand. En tegelijkertijd waren er ook nog die albumine-uitdagingen. Ik zal naar Joost kijken. De BSA-uitdagingen die we al een hele tijd hebben in het lab. Ja, er waren een aantal dingen. En dan moest je ook nog navigeren tussen 4 supervisoren. Wat ook niet altijd even makkelijk is. Dus ja, dat bracht soms wat twijfels met zich mee. Maar ik wist al die tijd wel dat je het in je had. Maar het was natuurlijk toch af en toe... Ja, het was even een beetje worstelen af en toe. Daarbij had ik ook nog, omdat ik wist dat je het in je had, gooide ik je af en toe een beetje in het diepe om te zorgen om je net wat meer uit te dagen. En dat was ook niet altijd even makkelijk, maar ik dacht, volgens mij gaat het er uiteindelijk helpen. En we hebben het er nog over gehad. Ik geloof dat het ook geholpen heeft. En ook om je te helpen met je zelfvertrouwen. En dat was echt precies wat je deed. Die groei heb je helemaal heel mooi doormaakt. Ja, gelukkig had je ook nog de steun aan Agnieszka en Rick in het lab, ga ik nu even zeggen. Die hebben je ook enorm geholpen daarin natuurlijk. En ja, de rest van het team en ja, iedereen hielp je er gelukkig doorheen. Waren daar op de momenten, je paranymfen ook, ja, de momenten dat je het nodig had. Er is er thuis ook nog een andere Rick, die ook heel belangrijk was. Samen met de rest van de familie natuurlijk, maar ja, vooral ook Rick, je levenspartner eigenlijk, hè. En jullie hebben samen een heel fijn thuis opgebouwd in een heel mooi appartement met de 2 vogels. Suki en, was het?
Momo.
Momo, zie je? Ja, ik wist niet meer zeker de tweede naam. Dus ja. Maar volgens mij een heel fijn thuis en heel vertrouwd waar je altijd op terug kan vallen. Dus dat is natuurlijk super. Ja, dan heb je naast je werk en in het lab had je dan ook nog, en je thuis, had je ook nog tal van passies. En ik was even bang dat Jos alles ging noemen al, maar ik had de taekwondo, het bakken, natuurlijk het haken wat we hebben gezien, ook in je boekje en wat hier staat. En ja, vooral dat laatste hebben we dus in je proefschrift terug kunnen vinden. Ja, en daarnaast de koekjes, wat Jos al zei, die creativiteit die was overal terug te vinden. In de social committee, maar ook in het organiseren van de fundraiser voor de Hartstichting, waar je heel mooie bedragen op hebt gehaald. We hebben nog een wandeling gedaan. Het heette een beachwalk voor de Hartstichting, maar het was een kelpenoler uiteindelijk. Dus dat maar ook heel mooi. Ja, en eigenlijk de hele tijd wist je en je werk samen met die passies op een hele unieke manier te combineren. Dus dat was natuurlijk heel mooi om te zien. Daarnaast hebben we ook nog verschillende uitstapjes gemaakt naar congressen en andere bijeenkomsten die deze periode extra bijzonder maakten. Van inspirerende bezoekjes aan Graz in Oostenrijk. Ben je ook nog naar Brussel geweest, naar Papendal, Groningen ben je nog naartoe gegaan. Waar we kennissen uitwisselden en ook nieuwe inzichten opdeden eigenlijk. En deze ervaringen hebben niet alleen jouw onderzoek verrijkt, maar die hebben je ook geholpen om je netwerk te verbreden natuurlijk. En ook daarin te groeien. Ja, en dan daarnaast vroeg je me ooit van, want ik zei altijd van, je bent heel slim. En dan zei je wel eens, wat bedoel je met slim? Waarom noem je mij slim? En toen dat vond ik een hele lastige vraag, omdat voor mij is dat heel vanzelfsprekend. Ik zie je als iemand die heel slim, intelligent is. Maar het heeft niet alleen te maken, denk ik, met de kennis. Maar het is vooral ook de manier hoe jij denkt en hoe jij de dingen benadert. En je denkt dan heel diep na, en dat hebben we net allemaal kunnen zien. Of het ging net eigenlijk vrij vanzelfsprekend. Maar je denkt over het algemeen heel diep na en dan zet je alles heel zorgvuldig op een rij. En dan vind je ook oplossingen waar anderen misschien blijven hangen. Dus ik denk dat je dat net heel duidelijk hebt laten zien. En dat voor mij was die definitie van slim, wat ik zie waar je heel goed in bent. Maar vandaag vieren we niet alleen jouw promotie, maar dus eigenlijk ook die enorme groei die je hebt meegemaakt deze periode. En je hebt niet alleen een prachtig proefschrift geschreven, waar je heel trots op mag zijn, maar je hebt ook jezelf eigenlijk beter leren kennen volgens mij. En jezelf ontwikkeld tot een zelfstandige, kritische en inspirerende wetenschapper. Dus voor de toekomst, ik wens je alle succes in de toekomst. Ik weet heel zeker dat je op een mooie plek terecht gaat komen, waar dat ook gaat zijn. En ik hoop dat we nog gewoon contact kunnen blijven houden en dat je in de toekomst hele mooie dingen nog gaat doen. Dus ook namens de rest van het promotieteam, gefeliciteerd.
Dankjewel.
Esteemed Dr. Willemars, dear Myrthe, it's my pleasure to congratulate you with your doctorate, and I do that also on behalf of Maastricht University. And I would like to share some impressions of the committee with you. First of all, you gave us a summary of your thesis, which I think was not only very understandable for us as a committee, but also, and that is meant for that, also for non-experts in the audience. Here, as well as in the live stream, I think that for most people became quite clear what you did the last couple of years in scientific work. And going to your thesis, I think it's a thesis on a very relevant topic. And it's indeed, as has been mentioned, the topic is in focus at the moment. And you did that in a well-written and very well-structured thesis. And this thesis was based, and also the publications, based on extensive and impressive work. Maybe not always came the right answer that you expected, but I think it's very worthwhile to do this type of studies. And I have also seen that a large part of the studies have already been published, and I think that most of it is underway or almost there to be published in scientific literature. With respect to your defense, we consider this as very strong. It is accurate, it was honest, and very structured. And we appreciate that you were very knowledgeable on quite a large spectrum of questions. And I think also that you have demonstrated that you are very capable to perform scientific studies of high quality. And so we are very satisfied with your defense. I do not only congratulate you with this result, but also your 4 supervisors, Professor Jan Glads, Dr. Miranda Nabbe, Dr. Joost Luiken, and Dr. Dietbert Neumann. Congratulations with the result that we have seen in terms of a very good thesis, an excellent defense, and a number of very good scientific publications, either already published or underway to be published. I would like to include in my congratulations also your family, your parents, your partner, and other members of the family, including your family-in-law. And I would like to congratulate you in particular with the results that you have seen in this academic ceremony, in which Myrthe was the center and the focus of this ceremony. I would like to include as well the audience here and the audience in the live stream, and I'm sure that quite a lot of other family members and acquaintances and colleagues have watched you today. I also would like to wish you a successful professional career and a fine and good, happy personal life. I would like to thank all members of both committees, the thesis assessment committee and this degree committee. Thank you for critically reading the manuscript. And Maastricht University appreciates your contributions, and in particular, I would like to include in my appreciation our external member, Professor Ventura from Paris. Thank you very much for contributing to this committee. Maastricht University appreciates it. I would like to announce that after this ceremony there is a reception. You find it very easily. We will leave as the first ones this ceremony, and we'll take a picture with you in the middle in the hall at the stairs in the hall. And I would like to invite the audience here to join us in the rafter for the reception to congratulate you. Hereby, I close this academic ceremony at Maastricht University.